Good day. In this video, we will be discussing about the different structures inside and on the surface of the cell. So what is a cell? So basically, the cell, the most basic definition is that it's the basic unit of living organism. So one good way to identify a cell is to name its components. So what are these components that are common to all types of cells? So the cell must have a plasma membrane. It's the membrane that separates the contents of the cell, the interior environment, from the exterior environment. It is a very special function because if this plasma membrane is broken, the cell literally dies. So the plasma membrane also controls the exchange between the cell and the environment, so that the interior of the cell will have a different concentration of solutes compared to the outside. So the cytoplasm is also another component that is common to all cells. So sometimes you call it the cytosol. It's a jelly-like mixture of water, sugars, ions, and proteins with all cellular components inside the cell. So basically, it's the fluid from which all the organelles, all the other parts of the cell are floating around inside the cell. So within the cytoplasm, some cells have membrane-bound organelles. Organelles are specialized compartments that carry out specialized functions. However, not all cells contain organelles. Now, all cells should also contain the hereditary material, which is the deoxyribonucleic acid or the DNA. So in some cells, the DNA is found inside an organelle called the nucleus, whereas other cells, they do not have membrane-bound nucleus, so the DNA is just in a region inside or floating around inside that inside their cytoplasm. So how big are the cells, or rather how small are your cells? So this here is a graph of the sizes. So here is let's start with the largest. So this is a tree, this is a whale, a giraffe. So a man is around 1 or 2 meters tall, and then going smaller, you have your duck. The range of sizes is the ants is around a few centimeters, and then when we count it in millimeters, we arrive at the eggs of the frogs. And then going smaller, most eukaryotic cells, the sizes are around in the hundreds of micrometers. So nasa range tayo with micrometers. And then bacterial cells, another type of cells, they are smaller. So they are around 10. So 10 to 100 times smaller, the bacterial cells compared to eukaryotic cells. So the cells have a really large range of sizes. And then going a bit smaller, you have your mitochondria, the size of the organelles. And then smaller than that are the viruses. So the viruses really are very, very small. They are in the range of nanometers. That's 10 raised to the negative 9th of a meter. Almost in the same range as your molecules, the biomolecules, which is 1 to 10 nanometers, that's the size range. And then going down, you have 0.1 nanometer, which are the small molecules such as the water molecules. So our cell is small, but not very small. So that is the relative size of your cells. So the cells are small enough that you cannot see it in the naked eye, but it's large enough that a simple light microscope can be used to observe it. So the cells are first observed in the microscope by Antony van Leeuwenhoek in 1665. So the most cells at that time that were observed at that time is around 10 to 20 micrometers. So they are very small, as I said. So we have your right now, the more powerful microscope, which is the electron microscopes, we can visualize the interior structures of the cell, its organelles. That's why we know a lot about the organelles now compared to before, because we can visualize them using electron microscope. And we also were able to find the existence of the viruses through the electron microscope, because the light microscope cannot be used to visualize viruses. Viruses can also be seen in the electron microscope. So just a brief introduction. So how small is the micrometer and the nanometer? So micrometer is 1 over 1 millionth of a meter, while nanometer is 1 over 1 billion. So that is the size range we are talking about. So these are several light and electron microscope image of the cell. So the one on the farthest on the left, this is just a light microscope. So in the light microscope, you observe it as a certain blobs, and then the organelles inside. This is actually, if I remember correctly, this is planaria. So you have blobs for the organelles. So from this vantage point, it's quite challenging to identify exactly what are those whitish blobs inside the cell. So another one is a light micrograph. So this one is polarized light. So we can see the surface, the surface of the cell. So this is a light microscope. We are looking at it see-through. This one is just the surface using polarized light. So you have bumps inside the surface of your cell. Now here is a fluorescence micrograph. A fluorescence micrograph is generated when you treat your samples with fluorescent dye. So the dye binds to certain molecules inside the cell. So you can have a plethora of other colors. For example, in this one you have two colors, yellows and reds. So the yellow one will bind to a certain protein, and then when you visualize it in a fluorescence microscope, the yellow dye will fluoresce. So you will identify which part which part of the cell that specific protein is located. So in here, I believe these are the asters of a certain cytoskeletal element. And then this is your transmission electron microscope. So here on the right are two images taken using an electron microscope. So we have two ways to do electron microscopy. You have transmission electron microscope and the scanning electron microscope. For the transmission electron microscope, you are going to generate only two-dimensional images, and you can view the interior of the cell as a sort of cross-section. So basically, when you take a TEM image, you fix the cell, and then you cut it cross-sectionally in order to properly visualize it. So this is a cross-sectional view. So you can see the interior showing your organelles. So these are the different organelles. It has a better resolution than our light microscope over here. In here, you just see smudges, whereas here, the organelles, it's very visible. So you can actually see packets inside. And then the other one is a scanning electron micrograph. So the scanning electron micrograph shows only the exterior of the cell. So you have the surface. And contrary to the light microscope, we cannot see those hair-like projections. So using the SEM, we were able to observe that your cell is actually hairy. So you have cilia. These are cilia. So it has hairy projections on the outer surface. So those are the different images on how you view the cells. So why cells are so small? So the reason for that is to maintain a low surface area to volume ratio. So there must only be a certain surface area to volume ratio for the cell. If you have a very low surface area to volume, so that means you have a small surface area and then a very large volume. There will be difficulties in the transport of materials in the interior of the cell. So that is why cells maintain having cells are small. So they want to have as much surface area as you can compared to their volume. So cells were first called cells by Robert Hooke in 1665, and it's considered the smallest unit of life. So we have here your cell theory. So the cell theory was the product of the different discoveries and theories proposed at that time. So we have several proponents of that. We have Matthias Kleinen, Theodor Schwann, and then there's also another one, Rudolf Virchow. So they were the ones that actually, because of their work, the cell theory was finalized. So what is the cell theory? The cell theory has 4 tenets. So one is every living organism consists of one or more cells. So the cell is the basic structural and functional unit of life. The cells are individually alive, even if you are part of a multicellular organism. That means you are you, but your cell is living, technically independent of you. So if you cut off your finger, the as long as there's still blood flow and there's still oxygen, the cells in the finger that you cut off is still technically alive. Although it will die off due to the lack of the oxygen because the blood now cannot properly flow. But anyway, so freshly cut, it's still alive. So all living cells arise by the division of pre-existing cells. So cells can only be made, or you cannot create cells from nothing. So cells come from pre-existing cells. So the cell contains hereditary material, which is the DNA, which they pass through to their offspring in the process of their reproduction. So how do cells reproduce? We have two ways, the mitosis and the meiosis. Although meiosis is a special type of cell division because this one is limited only for sex cells or sexually reproducing organisms. Now, let's move on to the types of the cells. So we have here the prokaryotic cells. So basically we have two major division of cells. So we have the prokaryotes and the eukaryotes. So in this video we will be discussing the prokaryotes. In the next part we will be discussing the eukaryotes. So prokaryotic cells are cells that have no nucleus, and they are the prokaryotic cells belong to the bacteria and archaea division or kingdoms. Kingdom bacteria and kingdom archaea. So they are single-celled organisms. So they are the smallest and most metabolically diverse life form that we know. So basically, it's like the structure of a prokaryotic cell is like a studio-type apartment. So you have the cell walls, you have your membranes, and then that's it. So the cell wall surrounds the plasma membrane. So your prokaryotic cells has cell wall because the cell membrane by itself is pretty fragile. So your cell wall is better protection for that. So it's made of peptidoglycans. So the molecules making up the cell wall are peptidoglycans. So that's peptides and carbohydrates. So that's for the bacteria, or proteins in archaea. So they are rather sticky. That's why the slime mold slimes, they are basically a community of prokaryotic cells. So they're very sticky. So they also have flagellum for motor movement. So this is a typical structure of your prokaryotic cell. So you have here the cytoplasm. So inside the cytoplasm, the ribosomes, the DNA, they have the DNA, and the other structures, the other proteins and molecules they needed to survive, they are floating around there. So that is your prokaryotic cell. So as I said, it's like a studio type apartment. Everything is in one room. So you have the plasma membrane, and then outside of that you have the cell wall. So you also have your pilus and your bacterial flagella. So your bacterial flagellum is for locomotion. Pilus can be used, the pili, they are hair-like projections, so they can be used to grab onto surfaces, or they can also be used for sexual, it's called sexual reproduction, but it's more of exchange of genetic information, because bacteria can perform sexual reproduction through exchange of genetic information. So other structures inside your cells, you have the ribosomes. The ribosomes are organelles that are used to create proteins. So without the proteins, the cells cannot live. So ribosomes is important for that. And then the DNA is on the nucleoid region. So they do not have a membrane-bound nucleus. Instead, there is a region inside where the concentration of the DNA is the highest. So that's the nucleoid region. And then we also have plasmids. Plasmids are also DNA. So they are small circular DNA that carry a few genes. Basically, when the bacterial cells do sexual reproduction or exchange of genetic information, the one that is being exchanged is actually the plasmids. So one plasmid to another. So this is also one of the reasons, the existence of the plasmid is also one of the reasons why we have your bacteria that are not susceptible to antibiotic or antibiotic resistant bacteria, because some antibiotic resistance is carried on the genes in the plasmids, and they can be freely exchanged between bacterial cells. So this is another view of your cell, prokaryotic cell. So that's it. So you have All the prokaryotes, before we end the topic about prokaryotes, so you have your prokaryotic cells are strictly single-celled organisms. They do not form complex organisms made up of multiple cells, so they're only single-celled. The closest they have of being multicellular is when they form biofilms. Biofilms are basically communities of your bacteria. They secrete layers of polysaccharides and glycoproteins, and then they use those polysaccharides and glycoproteins in order to build certain communities that are protected. So, for example, if you look at the bottom of a trash can that is slimy, that is full of slimes, basically that is a biofilm containing a community of organisms that, well, technically they together they secrete your polysaccharides and glycoproteins in order to protect themselves. So it's very difficult to remove them. Even dousing them in strong bases might not be as effective. So they can be bacteria, algae, fungi, protists, or archaea living in your biofilm. So here is an SEM image of a human fecal matter, and it's colorized, so you can see different types of bacteria living inside the fecal matter. So basically, your large intestine is a home to more than 30,000 species of bacteria. Actually, there has been an effort to identify those bacterial species. I think it's called the Microbiome Project. So because it's recently found that the existence of your microbiota can have indirect relation to your health, especially in the resistance to certain diseases. They can actually help resist or help your immune system in fighting off certain diseases. At the same time, if there is the bad bacteria inside, so that's actually one of the reasons for the good and the bad bacteria concepts that are found in the Yakult and other similar products. So you want to cultivate a good bacteria. If you have a bad bacteria inside your tummy or inside your large intestine, it might be a problem. So anyway, that's about for prokaryotes. So next, we will be discussing about the eukaryotes in the next part of this video.